In our last lecture, we discussed the central importance of proteins as the biological molecules that make things work in living systems through their roles as controllers of biochemical reactions, structural elements that hold parts of the cell together, motors that make things move, signals, and so forth. Let me remind you of two important take-home messages from that earlier discussion. First, remember that the function of a protein, what it does for the cell, depends almost entirely on its shape. The three-dimensional configuration of each protein determines its physical and chemical properties, which in turn allow the protein to serve its unique function. Second, remember that the three-dimensional shape of a protein depends almost entirely on the linear sequence of the building blocks, the amino acids that make up that protein. There are 20 different kinds of amino acids. An average protein might have a few hundred amino acids, and if you get the right sequence of those few hundred amino acids, the shape of the protein emerges. So proteins are the workhorses, their shapes determined by their sequence. How do we get a protein of a particular sequence built? Now, there are two questions here that we need to start addressing. Of course, very related. First is, what is the blueprint that we're going to use to build proteins physically made of? That is, what kind of molecule is it? And second, how does that blueprint, how does that molecule actually work to code information about the sequence of amino acids in a protein? It's the first question that we're going to address in today's lecture. What kind of molecule serves as the blueprint for making proteins? Now, on one hand, that question seems like it's pretty simple. But interestingly, that was one of the questions that really defined molecular biology in the 20th century. What we really want to know, and the big picture that we're aiming to go to here eventually, is how this blueprint acts as a code for making proteins. But before we could understand how the code works, we had to understand what the code itself is actually made of. And this is the smaller piece of the puzzle we want to understand next. Now, of course, today, everybody knows the simple answer to this question. We all know that the code is something that we'll call a gene. These genes are found on chromosomes. We've all heard this. And we also know that these genes somehow involve DNA. Now, these first two parts, that genes, or what we now call genes, were where the code resides, and more importantly, that genes somehow were on chromosomes, actually was already known at the very beginning of the 20th century. This was known from the work of early cell biologists. Walter Sutton and Theodore Boveri are actually two of the most important here, who discovered that the particular and somewhat peculiar movement of chromosomes that occurs when cells divide to make reproductive cells, cells that we call gametes, that are involved in sexual reproduction, that the movement of chromosomes during that event corresponded to patterns of transmission of traits between parents and their offspring. These patterns of trait transmission between parents and offspring actually had been discovered earlier by the Austrian monk Gregor Mendel, and we're going to talk about his work in just a couple of lectures. So they knew about Mendel's work, they saw how chromosomes moved, and they developed what's now called the chromosomal theory of inheritance, which basically means the way the chromosomes move is somehow related to the way that inheritance occurs, and therefore, following the concept that we're trying to develop here, somehow related to how information is stored. The information has to be on chromosomes. Okay, so we know that chromosomes are made of DNA, but how do we know that? And are they really made of DNA? Let me begin to explain how that became known by going back again to the earlier part of the 20th century to the work of an English physician named Frederick Griffith. Because this experiment that I'm about to describe really provided the first insight into how to get at the chemical nature of genetic information. Now, Griffith was a physician, and he wasn't interested in the molecular basis of inheritance or the molecular basis of genetics at all. Instead, he was addressing a much more applied problem. He was studying Streptococcus pneumoniae, which is a bacteria that, as its name suggests, causes pneumonia in humans. What Griffith wanted to do was develop a vaccine against this particular organism, because pneumonia caused by this organism actually often proved fatal. This was before the advent of antibiotics. Now, as often is the case for disease-causing bacteria, there were different strains of this particular organism that varied in their virulence. In other words, they varied in how likely they were to induce the disease and thus to cause death. Griffith was working with 2 strains of Streptococcus pneumoniae. And let me just name those for you. He was working with what we call the S strain on the one hand. This is a virulent strain. This is a deadly strain. Okay? It's called S because if you grow it in a colony on a little agar plate, it actually looks kind of shiny and smooth. So he called it the S strain, the shiny strain. And this is the deadly strain. And then he had another strain, which he called the R strain, which was non-virulent. If you got the R strain, you might be a little sick, but you wouldn't die. It wasn't a big deal. It's called the R strain because this strain of bacteria, if you grew it in a little colony, looks kind of rough on the surface. So it's called the R strain. Now, an important thing to note is that these strains of this bacteria did what we call breed true. In other words, as they reproduced, their offspring had the same properties as they did. In other words, S strain bacteria gave rise to more S strain bacteria, and so on. In other words, the difference between the S strain and the R strain bacteria somehow must be genetically encoded. So what did Griffith do? Well, Griffith was using the approach that was pioneered much earlier by Louis Pasteur, which was to take the organism that you wanted to develop a vaccine for and kill it so that it no longer could harm you, but nonetheless would perhaps induce some sort of immune response if injected, say, in a subject. So the idea was to take S-strained bacteria, kill them. And the way that Griffith did this, the common way to do this, is you just heat them up in solution and they all die. And then you take these dead S-strain bacteria and you inject them, say, into a laboratory mouse and see if that mouse develops immunity. The mouse wouldn't die if you injected S-strain bacteria that had been killed because they're dead. But the parts of the bacteria that are injected might nonetheless induce an immune response by a mechanism that we'll get to briefly very much later on in the course. This is a great idea, but it didn't work. And it often doesn't work. In other words, there wasn't enough left somehow of these dead S-strain bacteria to induce an immune response. So if you inject these in a mouse and then inject live S-strain bacteria, the mouse would die. It didn't become immune. Now, another common technique that was used then and now to develop immunity, to develop some sort of vaccine, was to inject not only the dead offending organism, but some related organism that was less virulent, in this case, the R-strain bacteria. The idea here is that the live R-strain bacteria, because they're alive, will induce the organism to develop a full immune response, but it's not going to kill the organism, and that development of a full immune response will somehow pick up some immunity to the dead S-strain bacteria. This is a common technique, and it often works. So what Griffith did then was he killed some S-strain bacteria and injected those S-strain bacteria in with living R-strain bacteria and saw what happened to the mouse. What he hoped would happen is that the mouse would develop immunity to the S-strain. But what happened instead was very unexpected and unfortunate for the mouse. The mouse died. Now, this is a surprising result when you think about it. It's highly unexpected because what's being injected into the mouse are dead S-strain bacteria that wouldn't kill the mouse and live R-strain bacteria that wouldn't kill the mouse. So nothing was injected in the mouse that should kill it, and yet the mouse died. What Griffith found out when he dissected the mouse was that inside the poor dead mouse were living S-strain bacteria. He injected dead S-strain, and when he took the mouse apart,